Thank you, Stan. Great pleasure to be here. Thanks for organizing this wonderful meeting. Um, Today, I'm not going to present to you a brand new technology, but instead I'm going to use brand new uses of a very old technology. If I may be able to advance the slides, I'll be delighted. The right button doesn't work. Left button, maybe. No. That's okay. Now turned on. Okay. Speaking of an old technology, intracranial electrophysiology, as my friend Florian very nicely described, has been in use for decades, and we have talked about its pros and cons. It is, of course, as any other tool, has its own limitations. This is a technology that started with pioneers' work in Europe with Talarak and also in Northern America with Penfield and Jasper. Uh, what they were not able to do was to really decode the signal that they were recording from because of the amplifiers they were using and lack of computations and sampling rate, etc. Therefore, this was the bandwidth that Penfield had to work with. Hence, Penfield called intracranial EEG useless. And instead, he was very interested in electrical stimulation for the study of human mind. Those days, this was how the signal seemed to them. But now today, if you really open up the sampling rate and go higher up, you see a flurry of beautiful brain signal that is generated as human beings are engaged in specific cognitive tasks. So now, one advantage of this method is that it's done in humans and it takes very little training. You can do it with very simple instruction. And also, the most important thing is that humans, unlike animals, they can talk and they can tell you what exactly is going on. They can share their subjective feelings, something that remarkably was harnessed by Penfield's electrical stimulation work and their amazing description of what patients reported. And of note, Penfield was Sherrington's student, and as you know, Penfield not discovered, but described the somatotopic organization in the sensory cortex. The motor cortex was done by Sherrington because he could do it in monkeys and show somatotopic organization. But he was unable to map the sensory cortex because monkeys couldn't tell which part of the finger is tingling when Sherrington was stimulating. Something that Penfield very nicely tapped into and described the somatotopic organization of the sensory motor cortices. Something that we can do now is to just stimulate a specific region of the brain and then discover what exactly happens to the human mind. And you can then tie it into functional imaging work that's done in the last several decades that Dr. Dehan very nicely summarized. Every corner of the brain, you stimulate something very specific, always is reproducible. Something very specific is reportable by these human subjects. Again, cross-subject consistency is remarkable when we electrically stimulate. It is not the case that when you stimulate the, let's say, temporal lobe in one patient, you get some psychic reports, and in another patient, you get a motor report. It's always consistent, although the details may vary a little bit, but the nature of the subjectivity induced by electrical stimulation of a specific part of the brain has been always consistent. What we have recently discovered is that certain areas of the brain are always silent when you stimulate. Certain areas are hot. And we mapped it to human connectome data, and we discovered that areas that are dark, here you see in darker color, versus areas that are hot, that you are seeing in bright red, are very distinct in terms of which network they belong to. And the darkest one that you see here is the default mode. And I will show you later an explanation for this. But there are certain areas that remain always dark. And those are the ones that are highest association cortices with most diffuse connectivity patterns. The ones that have less diffuse connectivity patterns, the ones that are closer to the sensory motor inlets, outlets, are the ones that are usually very hot. Another thing we can do with this method is to stimulate repeatedly with single pulses in one region of the brain and then record evoked potentials in certain areas. And we can also compare, for example, the presence or absence of these evoked responses and deduce that area A is connected with area B functionally and effectively and causally. And we can also compare the speed, we can compare the strength, we can compare the sturdiness of this connectivity and put together with sophisticated machine learning algorithms to come up with clustering algorithms to come up with some profile of connectivity in the large human brain, something that has not been possible before. So today I will try to use one example of this combined electrical stimulation recording method. And I'm going to focus on the study of sense of self. I have boldened the term sense because truly this is about the sensing of subjective phenomenon of being present, knowing that it is you. It is the sensing element. It is not a very higher order, entirely abstract type of phenomena such as soul or anything of that sort. I'm just going to talk something very simple, which has to do with the sense of subjectivity. Here, I will borrow some terms from James in terms of me and I and how these can be actually tied to the sense of subjectivity. My take-home message is that this problem can probably have much simpler solution than we have been debating about. Happy to hear your critical comments on that. My take is, again, my perspective of a clinician. I'm studying patients with epilepsy. You might ask, well, what grounds I'm standing on? Are those shaky grounds? Are these epilepsy patients okay models for studying the human brain? I just remind you that patient HM was an epileptic patient, and discoveries from his brain led to tens of thousands of publications about the hippocampus and memory. Also, somatotopic organization was done in patients with epilepsy. Lateralization of function was also from patients with epilepsy. So I will be happy to discuss at length. It is a misunderstanding to think that epileptic brains are miswired. As Florian very nicely alluded to, these patients we do recording from suffer from focal epilepsy. So there's only one network that's affected. The rest of the brain is unaffected. And even in the pathological tissue, we have shown that you can actually get normal responses as long as there's no epileptic explosion happening. So the story begins with subject S19137. Couple of minutes of video. This is what he described to me in the office, and that led to some really interesting findings down the road in the next few slides. Let's see what the patient reports. He's reporting what happens to a human mind when epileptic activity occurs in a specific network of his brain. He's a very highly functioning person. He was very articulate, as you will notice. And here he describes a slow seizure. Can we get more volume, please?
One of the most interesting experiences of my life. It was kind of like if you imagine taking my seizure and just stretching it. It was not more. It was not more.
He's talking about the seizure that was slow, that got kind of stretched, so it gave him an opportunity to know what exactly is going on in his mind.
So the first thing I noticed was that it felt like the whole world, like again, these 6 degrees of freedom I was talking about, sort of the three spatial dimensions, and then there's theta x, theta y, theta z. I could feel them move, and then the next thing I knew is I was getting, I would say, the emotion or feeling of where in this 3D space am I? And I went through the process very slowly of refiguring that out, but it was like listening to another conversation in my brain. Tell me more. It was really, I really enjoyed it. It was kind of like, experiencing. It's like okay, there are. I'm just going to describe it as a computer because that's my best way to describe it.
Yeah, sure.
Imagine there's these two ICs speaking with each other, right? These two components, and they speak with each other, right? But there's one data bus. Are you familiar with the data bus?
I wish I were familiar. No.
What it does is there's this one information highway, and what it does is it says I want to speak with this component X, Y, Z, A, B, C, D, right? Now A and B are speaking, but they can only speak amongst this information highway that the CPU is always able to listen to. So I'm listening to A and B speak with each other, trying to figure out what the hell is going on, and as a result, I'm listening to this conversation. The CPU is trying to figure out, maybe that I'm me, I don't know what that means necessarily. I personally I don't believe the idea of a soul personally. But, I was aware that I was listening to two parts of my brain speak to each other in a way where this third part of my brain, which I considered me, was able to listen. And what that felt like was this. When they talked about depersonalization, it was,
Can you stop the movie?
My friend asked me something recently. He said, what would it feel like if some...
Oh, anyway. So I wanted to stop there and emphasize what he said. He said, I was part of a conversation in my brain that it wasn't me. I was observing parts of me talking to each other that I wasn't part of. And I will show you what exactly happens when we electrically stimulate that area. But a patient comes in with seizures like this. What we usually do, we do a structural MRI to see where the structural lesion explaining epilepsy might be. He was non-lesional. We do FDG PET to look for hypometabolism. He had none. Scalp EEG to see where the electrical discharges are. They were primarily in the midline region in the parietal areas. And video EEG monitoring of seizures suggested that, again, seizures are coming somewhere from the posterior quadrant. We didn't know left or right. Psychometric evaluation was not that helpful. So in these cases, we have a hypothesis that's coming from the posterior quadrant and midline areas. So we put these electrodes. These are clinically mandated. It's not like I decide 20 electrodes. I can put it anywhere I like, especially in the United States with liabilities and legal lawsuits, et cetera. We have to be very careful. This is decided in a consensus meeting among radiologists, psychiatrists, psychologists, neurologists, and neurosurgeons. And lo and behold, we found the focus, and it was on the right precuneus. As you see, now we have both left electrode and right electrode. Left one is so-called healthy, right one is epileptic. And now we go and stimulate the homotopical area on the left side, and this is what the patient reports. What you created with the stimulation was that separation. This is, you can change that, these are patients' own words, but you can change it to dissociation, and we get into self-dissociation in a second. The same way a pilot can lose control of a plane. They can be forced out of the cockpit or forced or not control it, but still see what's happening to the whole plane. That is what just happened. I got pulled out of the cockpit, or I got pulled out of the chair, the pilot chair, but I could still see all the gauges. When the subject was asked if he was floating in space, he said, no, I was floating in myself. Okay, so this is a clinical observation we are making in a patient, single subject. You might argue, well, that's the epileptic tissue on the right side. Maybe the left side is also epileptic. So we gathered more subjects throughout about 2 years. And now we have 8 subjects with electrodes in a similar area that is not epileptic at all. Now, these 9 subjects together, including the last subject, you see the location of the so-called hot areas that are causing similar phenomena is in the right precuneus. This area is quite hot. A lot of literature on this, but what I ask you to be careful about is that in the literature you hear many different names referring to this neighborhood. Sometimes we call it posterior cingulate, sometimes retrosplenial, sometimes precuneus. And as I mentioned, it's in the literature is hot, but also in biological terms, it's very hot. It uses about 30% more glucose at rest than the rest of the brain, partly because it receives both anterior and posterior circulation blood flow. So you can't really cause a very focal injury such as stroke by just affecting one area. Thanks to Stephen Lorry's work, this was shown to be one of the major areas that is affected in vegetative state in terms of abnormal function compared to the rest. That might be related to what I just described there in terms of the blood flow. It's also shown to be one of those areas that first gets kind of affected when you put anesthesia. You have heard a lot about this particular postremedial cortex in the context of default mode that Uri Hassan has very nicely lately described as the making sense network. This is going to be quite relevant to my conclusion of this talk, what I'm trying to make sense of what I presented to you, no pun intended. So here, just to review the anatomy on the right side, you see that precuneus is dorsal to the splenial sulcus. And anything that's ventral, including posterior cingulate cortex and retrosplenial cortex, are not part of precuneus. Precuneus anterior has evolved quite a lot in human and also primate brain. On the left side, you see the anterior precuneus has almost like a shape like an S in the sagittal. And the belly of that S is area when you stimulate, you cause dissociation. It's as specific as it is. Now, this is showing the specificity, anatomical specificity. We stimulated every single electrode. Nothing happens until you get to that pair of electrodes that we stimulate and see what the subject reports. See if it works.
You're not feeling anything? No, I haven't done it yet. 1, 2, 3. That was crazy. Tell us more.
Do it again.
1, 2,
I'm
3.
pretending I'm stimulating, even saying 1, 2, 3.
How about now?
That's
1,
the sham.
2, 3.
No
How about
electricity
now?
is delivered.
1, 2, 3.
Oh, yeah. Kind of like my body lifted out.
Lift it up.
More on the left side. Kind of like I lifted out like a little bit.
Who was lifted?
Like, kind of felt like my soul was leaving my body.
Oh, actually, it's very...
All right, I won't go. It's a 5-6 minutes video. She describes this phenomenon that we see in these subjects very reliably. We stimulated repeatedly the hot electrodes that are causing self-dissociative symptoms. And we observed on the left side, you are seeing the outflow where the signal goes to. because I'm not a phrenologist. None of us is. I don't believe that the anterior precuneus is seat of anything. I do believe in a system of interconnected structures that are working together. That's the outflow. And then here on the right side are areas of the brain that project to this particular hot zone. And you see on the top is the hot, in the middle is the cold ones in the precuneus. And here is a summary of it. We then took the patient's fMRI data and also human connectomic data. It's very hard to interpret these in small scale here, but what I can assure you is that what we found was that this particular hot zone is not part of default mode, whereas the cold electrodes were all part of default mode. And that fits very well with series of studies we have done. When we stimulate the default mode, nothing happens. As you go further dorsal or ventral, you get a lot of senses reported, but when you are in the ventral posterior cingulate or retrosplenial, nothing happens. And that goes with what I explained to you, that it's part of the darker zone. And when you look at the human connectomic data, those darker areas correlate with the degree of diffuseness of their connectivity. The higher diffusion, the higher associative area, the higher hierarchy, the less they can actually induce any subjective change when you tickle them. So what about this story of me and I? We are borrowing some Jamesian language here, and we are putting a lot of weight on it based on this uh, preliminary observation. But the idea is as follows. And this is just used as an example to you guys, showing how clinical observations from one patient leads to 8 more patients, comes up with a hypothesis like this, and then can be tested, hopefully with some pragmatic value at the end for society, for the sake of society, in terms of alleviating suffering of patients. The way that we explain it is that default mode is is the associative network that has to do with retrieving knowledge we have gained through our experience and through our exposure and culture, etc., societal interactions. That is the extended me, meaning whatever knowledge I have about the world and also my experience in that world. It is the object. Whereas the network that anterior precuneus is part of, and something I didn't detail, it actually goes to areas of the brain that are associated with visuospatial, vestibular, and proprioceptive body envelope information processing, is the agent, is the experiencer, is the subject. And that's why we use the term I. And this goes very well with pioneers such as Olaf, who's sitting here, who has talked about multisensory mechanisms for embodied self and self-consciousness. And the way that we describe it is that these two systems are in real time and millisecond resolution, are interacting with each other. When I'm experiencing the world, I retrieve memories that are relevant, but I also have to have that stamp of subjectivity of me experiencing the world. We think the I system provides the point of, or the perspective, the point of view, whereas the me provides the content that's relevant to that experience. And when we stimulate that, we cause some sort of a dysfunction or disjointment of these two systems, where the pilot is no longer the pilot, but the pilot sees what's going on because of the conceptual knowledge of, I know I'm in this hospital bed, I know what to predict, what should be happening, but that's not what the subject is experiencing, hence the dissociation. So going to a little bit philosophy based on these observations, and brain science comes to help philosophy. We think Descartes was not very right. We think the opposite might be the case, that we do have the I that enables thinking rather than the other way around. So, last word before I finish, you might ask, okay, what about theories of consciousness proposed by others when they bring in the sense of visceral sensations and interoception? We do believe that they are very important for emotional coloring of the experience, but they may not be associated with this sense of self that we think is extremely important and very highly temporally resolute. Last, what is the value of these type of observations? Some of these patients have been exposed to intravenous ketamine as part of our collaborative research with Carl Dieserath. And they explained that what we did with electrical stimulation, at least 3 of the 8, explained that this was very similar to what they experienced when they got IV ketamine. And that brings me, and this has also been reported by Carl in a collaborative work. He did the study with rodents and found that the postremedial cortex was one of the key regions showing pathological oscillations after IV ketamine in rodents. And these rodents had the phenotype of self-dissociative behavior. What we think this could be related to Helen Mayber's pioneering work in depression. We think that this could potentially be related to the effect of psychedelics that Dr. Knott and others have studied. This could potentially be an area for neuromodulation where we can actually detach the self a little bit, because we believe that depression has to do with self-ruminative process, too much of self, too much of that stuck in the vicious circle of thinking about self and relating everything to self. And precuneus could potentially be a relevant target. Lastly, we heard about the blood-brain barrier. I'm a clinician. I do believe that neuromodulation is the way to go for the brain, because unfortunately, because of blood-brain barrier, we are dumping a kilo of pharmacological agents into the blood. 900 grams of it goes to flesh and causes all sorts of side effects in the liver and pancreas. 10 grams of it passes the blood-brain barrier. 9 of it goes to parietal and occipital cortex and causes all sorts of problems with diplopia and sense of instability. And 1 gram of it goes to the circuit that it's supposed to go. I'm hoping that one day we will identify these circuitries and we will apply side effect-free neuromodulation. And that is where I like to end my talk. So thank you very much for your attention.